with my talk. This is entitled NMR and MS-based micrometabolic profiling of special plant cell types and tissue. So as announced yesterday already, in our institute we are interested in the localization of secondary metabolites in special plant tissue. And as you certainly know, metabolites are not evenly distributed in the organisms, in the plants, for example, but they accumulate in certain specialized tissue and cells. For example, they may accumulate in trichomes on the surface of leaves, in root hairs or on root tips. They also may accumulate within special secretory cells within tissue, or they are also sometimes found around in certain specialized cells of the leaf, and so on. We are looking for such compounds in order to establish their importance in ecological interactions. And the methods we are using, and which I will present here, are imaging techniques. First of all, multi-imaging, and also we are using NMR to detect and also to quantify compounds by inside these cells after dissection using laser technologies. So the first example which I will present to you is about banana plants interaction with nematodes. We have heard in the previous talk nice data about nematodes. I will talk about ecological interactions of nematodes with the banana plant, and I am talking about the Rhodophilus similis nematode. This is a roundworm living in the soil and attacking the roots of the banana plants, and after some time they are toppling, and they get unstable, are toppling, and of course this is connected to loss of the harvest of the fruits. And what we know is that in the roots of the banana plants there are some special defense compounds accumulating. They belong to the phenylphenolene type and the oxidized phenylphenolenones. And here in this project we were using susceptible variety and a resistant variety, and incubated these two varieties with nematodes, and then we observed differences in the accumulation of the products within these roots. First of all by multi-mass spectrometry. Now this is just a figure how it works. We have a sample which is placed or mounted on a multi-target, and then it is sprayed with a matrix in order to improve ionization after laser pulse on the surface. And then point by point there's a matrix irradiated point by point with a laser. Mass spectra are recorded and reconstructed by computer programs to produce an image displaying the distribution of certain compounds within a certain area, for example in a leaf or in a petal or whatever it is. And this method can also be there's a shortcut using the same method or similar method without the matrix for some compounds. Aromatic compound, it works quite well without a matrix, and we used this technique to detect compounds within these lesions caused by nematodes in the susceptible and in the resistant variety of the Musa plant. And here you can see some masses detected which correspond to the compounds which you can see here. In some cases there are isomers, so that we are not sure in each of these cases, but in most cases it's quite clear that we have accumulation of these compounds in the specified lesions in the tissue. And another part of the project was about the fate of these compounds within the nematode. So the nematodes are killed by after digesting these roots, and we found that within the nematodes there are lipid droplets which are fusing after digestion And this is a typical dead nematode. This is not curled. It's a straight dead nematode, and by using laser desorption mass spectrometric imaging, we could detect these compounds within the lipid droplets. The lipids have been detected by Raman spectroscopy, just to mention that it is really a lipid droplet within the nematode. The next projects are using laser microdissection to sample certain tissue and plant cells. There was a paper in 2005 by people from Golm, in Max Planck Institute in Golm, and they have been reported that they cut vascular bundles from Arabidopsis plants, and then they recorded mass spectra and detected a number of primary metabolites within these sugars and other amino acids and so on within these vascular bundles. And we were using a similar technique, cutting or using the laser to cut certain cells or tissue pieces from cross sections or from cryosections or just from surface material, and collecting these cut materials, sonifying them, and then subjecting to NMR and to mass spectrometry. And the projects which I will show you in this context is about rapeseed, and another one is about trichomes from two plants, cannabis plant and the wild tobacco plant. So the rapeseed is important because of this oil production, but it is also famous for its glucosinolate contents, which provides the typical taste of the brassica plants. And there are a number of other secondary metabolites within the plant and also within the seed. And what a PhD student of mine has done is to cut the seed tissue, cryosections of the seeds into several parts, and then extract the tissue pieces and record mass spectra, and we were able to show that there is an accumulation of flavonoids and some spermidine conjugates within certain parts of the tissue. Glucosinolates are almost evenly distributed except the shell, and also some phenylpropanoids or some synapine conjugates. They are distributed almost evenly except the shell, but the phenylpropanoids, camphorol glucosides, and also the spermidine conjugates, they are actually accumulating in some tissue, hypoglycemic tissue, in the radicle of the seed shell of the seed. Another project, as I mentioned, is about trichome detection of metabolites in trichomes. First of all, we tried to use a wild tobacco plant, which is a mutant which has large trichomes, and we tried to detect nicotine and other compounds by magnetic resonance imaging, but actually the sensitivity of the imaging is not sufficient to detect metabolites. So we went back to extraction and detecting the metabolites by NMR spectroscopy, by proton NMR, just by proton NMR, and comparing the compounds or the spectra which we detected with the spectra of reference material of nicotine. And in this case, it was unknown that this compound is occurring in the tobacco plant, so we have been isolating a lot more and then comparing the isolated compound with the spectra of the compound cut from the trichomes. So these spectra have been recorded at 500 megahertz spectrometer with a cryoprobe in methanol. Another project was together with Oliver Kaiser group about trichomes of cannabis plants, and we here in this case some parts of the trichomes, this head of the and the stem were separated by the laser and again extracted and NMR spectra recorded. The same procedure, proton NMR spectra compared with reference compounds, and this led to identification of several cannabinoids within different type of trichomes on the surface of the cannabis leaf. So final part is about two further projects, laser microdissection combined with NMR to detect some phenylphenanones in secretory cavities of a phenylphenanone producing plant, and laser microdissection combined with NMR in Norway spruce, in the bark of Norway spruce. This is about the dilatris plant, a Hematoaceae, that means blood root, because the roots of these plants are colorful according to the occurrence of phenylphenanones. But also the leaves do contain some of these compounds, and there are special secretory cells, and we were using the laser to dissect these secretory cells. This is the workflow. So the laser is first of all, the instrument is used to mark the track. Then the laser is cutting, and this is the dissected cell, and this is the hole after remaining after the remaining hole after the cell has been cut. In this case it was a quite large cell, so it is not a normal cell. The diameter of the cell is just 60 to 80 micrometers in some cases, and it was sufficient to collect around 8 to 10 cells in order to receive some very nice NMR spectra like this with a good signal to noise. And by comparing again with reference compounds from the same plant material isolated before, we were able to identify 3 of these compounds and also to quantify the compounds by means of integrating some specific proton signals. This is shown here, so the ratio finally was determined to be 11 to 7 to 2 between these 3 metabolites. Norway spruce is my last project which I'm going to present here today. Norway spruce is endangered by the attack of the bark beetle, which is associated with a fungus, Ips typographiscus, which actually finally kills the tree. And after infection by the fungus, the tissue, the bark tissue, starts to change. This is shown by this fluorescence image here, microscopic image, and it shows that there are several different type of cells which have different fluorescence. And of course, we were interested what happens in this bark tissue and what is the contents of these specific cells. And so we were cutting 2 type of cells, these so-called stone cells, and also the fluorine parenchyma cells, which occur in rows and which are filled with some sticky material upon infection. And we also cut some control tissue. And this is again the procedure. These fluorine parenchyma cells were first identified in the microscope. Then the laser is used to cut some cells, and this material then is subjected to extraction and to NMR and mass spectrometry. And some sieve cells were collected as a control to show the difference between the contents of the PP cells and the sieve cells. This is again a picture of the tissue. We identified these parenchyma cells by means of some starch grains which are inside, and which clearly indicate that we are cutting the correct cells. And the same procedure again, using this material to record proton NMR spectra and to compare different type of cells with control materials and with reference compounds, and we were able to identify stilbene astringine, which is a very strong antifungal compound within the bark tissue, and also catechin was present there. Of course, and in this case it was interesting, we used some contaminations for for quantification, so the contamination was always present in the same concentration, so we integrated this against the signals of catechin and astringine and were able to quantify the material. The same procedure was done with stone cells, and in this case we again did NMR, proton NMR, and comparison with reference compounds. And it's interesting that we, after some collection of some material around a milligram or a little bit more, we were able to see this nice proton spectra, and even we succeeded in recording some COSY and HSQC 2D NMR spectra to make sure that even without reference these compounds are coming up in the tissue after infection. Okay, that's the talk, and this is the group again, as I showed yesterday. In this case I have my special thanks are due to Ecology, who set up the laser microdissection in the institute, and Jingjing Fang, who has done the work with the glucosinolates in rape. Yeah, and there's our collaborators. The Spruce project was done with some people from Norway, in Oslo, or near Oslo, Arsh near Oslo, and the project about cannabis was done together with Oliver Kaiser. Sheng Hong Li is now in Kunming Institute of Botany in China as a professor, and he also was contributing to the project with the Norway Spruce, and Alex Swathurst is always collaborating with us in terms of mass spectrometric analysis, and these organizations give money for different projects. We have seen no difference, no changes actually by the laser, by the action of the laser. There is no decomposition, but we cannot exclude with other type of materials. If there are sensitive compounds, probably there may be some changes. And the laser technique is also used frequently to collect material for RNA analysis, and even in this case it works quite well, so there is no serious decomposition. Relative to extraction of whole material, of the whole leaf material, for example. So we see the same compounds in the whole leaf after extraction. Yeah. And we actually used the control experiments in order to get some material as reference compounds.